Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today we will discuss from question number 31. The base pair stack is apart. Option A 0.24 nanometer, option B 0.34 nanometer, option C 0.35 nanometer, option D 0.36 nanometer. So the correct option is option B 0.34 nanometer. Question number 32. The model of DNA replication in which one strand is conserved and the other is synthesized is called option A conservative, option B semi-conservative, option C dispersive, option D none of the above. So the correct option is option B semi-conservative. Question number 33. In semi-conservative model, the strands are separated and act as option A enzyme, option B tool, option C model, option D fingerprints. So the correct option is option C model. Question number 34. The model in which both strands are preserved is called option A conservative model, option B semi-conservative, option C dispersive, option D none of the above. So the correct option is option A conservative. Question number 35. The model in which both strands break is called option A dispersive, option B conservative, option C semi-conservative, option D none of the above. So the correct option is option A dispersive. Question number 36. The semi-conservative model of DNA was proved by option A P.A. Levin, option B Maselson-Stahl, option C F. Meister, option D Watson and Crick. So the correct option is option B Maselson-Stahl. Question number 37. Maselson and Stahl used radioactive isotope of option A carbon, option B phosphorus, option C sulfur, option D nitrogen. So the correct option is option D nitrogen. Question number 38. The enzyme which attaches the fragments of DNA is called option A DNA polymerase 1, option B primase, option C ligase, option D DNA polymerase 2. So the correct option is option C ligase. The enzyme which attaches the fragments of DNA is ligase. Question number 39. The true replicating enzyme is option A DNA polymerase 3, option B ligase, option C primase, option D DNA polymerase 2. So the correct option is option A DNA polymerase 3 is a true replicating enzyme. Question number 40. The enzyme which plays a supporting role in DNA replication is option A DNA polymerase 1, option B primase, option C ligase, option D DNA polymerase 2. So the correct option is option A DNA polymerase 1. Question number 41. The DNA polymerase adds how many nucleotides to the end? Option A 2, option B 3, option C 4, option D 5. So the correct option is option B 3. Question number 42. The strand which elongate towards replication fork is option A leading strand, option B lagging strand, option C both A and B, option D none of the above. So the correct option is option A leading strand. Question number 43. The length of Okazaki fragments is option A 100 to 200 nucleotide, option B 200 to 300 nucleotide, option C 300 to 400 nucleotide, option D 400 to 500 nucleotide. So the correct option is option A 100 to 200 nucleotide. Question number 44. Okazaki fragment is a option A RNA, option B DNA, option C protein and option D enzyme. So the correct option is option B DNA. Question number 45. Okazaki fragments are synthesized by option A DNA polymerase 1, option B primase, option C ligase and option D DNA polymerase 3. So the correct option is option D DNA polymerase 3. सो डियर स्टूडेंट्स, ये था हमारा आज का लेक्चर। कल एक न्यू वीडियो के साथ मिलते हैं। स्टे कनेक्टेड। अल्लाह हाफिज़।